In this video, we are going to start to discuss the structures of solid materials. The structures of solids primarily determine their properties. This is why it's important to have a basic understanding of the different types of structures for solid materials. For example, we can use mechanical milling to access sodium, lithium, and magnesium. That makes this metastable rock salt, which is then used in manufacturing. Wafers and wafer technology relies on understanding how silicon atoms can be used to form a semiconductor. We've used X-ray crystallography to understand the structure of DNA, and the fact that it's a double helix is actually what makes it such a robust, proofreadable way to store genetic information. And the beautiful six-pointed ends of snowflakes is just because of the orientation of water molecules in a crystal structure of ice. So there are two main types of solids. We've got crystalline and amorphous. So a crystalline solid has a regular ordered structure, and we will talk more about this in a little bit. While an amorphous solid doesn't really have a long-range order, and you can see this difference in quartz and glass. So quartz and glass are both made up of oxygen and silicon atoms. However, in quartz, there is a regular repeating pattern to the structure of the quartz itself. And whereas with glass, although there is a regular repeating pattern of the way that the atoms are connected, you can see that they are kind of connected in a different way. So yes, you have silicon, oxygen alternating, but you'll have some larger and smaller sections. And so it is called an amorphous solid because we don't necessarily know what the precise order is of this material. So I'm going to talk very briefly about X-ray crystallography. You do not need to know how to do any major calculations or anything like that, but it's important that you at least understand what it helps us know about things and where the technology kind of comes from. So here's the basic outline of how we use X-ray crystallography. First, we crystallize something. So usually we use this to determine protein structure. And so we'll take a protein, a solution of protein in a small container and either add a seed crystal or scratch. You can let some crystals of this protein grow, and then you put it into the X-ray crystallographer. You look at the diffraction pattern, and then you do the computer does calculations, looks at the electron density map, and you can create a protein model, or the computer can create the protein model. So this is similar to the way a prism separates component wavelengths of light, because remember, X-rays are electromagnetic radiation, just like visible light is. They behave in the same way. It's just that the wavelength of X-rays is much shorter. They have much higher energy. And so they have this smaller wavelength. And with light, with visible light, or with an emission spectrum, we see that we separate into these colors. And so what happens is if you have refraction, that's when light will bend. So this refraction is why if you put like a pencil in a cup of water, it looks like it bent. Reflection is just bouncing right back, and diffraction is splitting of the wavelength into smaller subsets. So diffraction is what's happening in X-ray crystallography to give us a pattern that can let us look at a structure. So this is what it actually looks like. So you have an X-ray tube source. It passes through a lead screen and hits your crystalline solid. So you have a beam of a focused beam of X-ray light hitting the crystalline solid. And then the X-rays diffract in different ways based on the shape of what's in the crystalline solid. And so the X-ray detector gets this image, and then that is analyzed later. And here is how it actually works. So X-rays can penetrate that first atomic layer and there is an angle here, and they measure that angle. So light is bent according to the distance between those atomic layers. So this works because X-rays, a form of electromagnetic radiation, also behave like those light waves that we saw earlier in the chapter on the atom. And constructive interference occurs when two waves interact with their crests and troughs in alignment, so you get a double amplitude. And destructive interference occurs when two waves interact with the crests of one aligning with the troughs of the other, so you get like a flat line. So that's destructive interference. And then, so when light encounters two slits separated by a certain distance, the constructive and destructive interference produces a characteristic pattern. Okay, so we saw this when we looked at the weird behavior of electrons earlier. So the thing is that the atomic planes within crystals are separated by about 100 picometers, which happens to be a similar wavelength to an X-ray. And so the interference or diffraction pattern is seen when these X-rays interact with the distances between those atomic layers. Now, your textbook goes into some detail about how these angles are calculated. You do not need to learn how to do that or perform calculations with this. But what I do want you to know is what X-ray crystallography is primarily used for, and that is to determine the structures of simple atomic lattices as well as structures of proteins, DNA, other biologically important molecules. The most famous of which is the image taken by Rosalind Franklin that helped Watson and Crick and Maurice Wilkins figure out what the structure of DNA was. And it was that alternating pattern of constructive and destructive interference that showed them that it was likely a twisted lattice structure. I'm sorry. That likely showed them that it was a twisted helix structure. Crystallography has also been really important in medicine. So for example, without X-ray crystallography, we probably wouldn't have been able to develop our HIV drugs as quickly as we could. It was learning what the shape of those HIV enzymes were that allowed us to generate drugs that would specifically bind to those proteins or to those enzymes or to those compounds and inactivate them. So a crystalline lattice is nature's way of minimizing energy. And so we represent lattices with a unit cell, and you saw this with lab. The unit cell is just repeated over and over and over at the entire lattice. So in this case, our unit cell is this pink box here, and it gets repeated throughout the structure. So this is a 2D representation, but we could imagine that if this was a 3D representation, it would just be a cube, and it would be the same. This would be the same on all four sides, or all 6 sides, sorry. So here we see that there's 1 in the middle, and then there's a quarter on the outside. And so this unit cell will repeat. And what we're going to do is focus on these unit cells. So a cubic unit cell has equal edge angles and equal edge lengths, and also angles of 90 degrees. So it would look like this. And in this section, we are mostly just going to focus on cubic cells. That is what is the focus of a textbook. And you don't need to look at the rest of these. However, if you are interested in crystal structures, especially geology, you might find these really interesting to look at. So to start off by classifying these cubic structures, we're going to look at the number of atoms within each cell and the number of atoms that in which each atom is in direct contact. So these are the 2 things that we care about really. So the number of atoms in which each atom is in direct contact is the coordination number. And the larger the coordination number, usually leads to higher forces holding these things together. And a simple cubic cell has a coordination number of 6. The packing efficiency is the percent of the unit cell occupied by spheres. So the larger the coordination number, the larger the efficiency. So this simple cubic cell has a packing efficiency of 52%, meaning that only 52% of this cube is occupied by these purple parts here that represent particles, and the rest is empty space. So each corner is 1/8 of a sphere. So if you were to count up all these spheres, we see we have 1, 2, 3, 4, 5, 6, 7, 8. So we have 8/8. So that roughly works out to being 1 atom's worth in this simple cubic unit cell. So we're going to go through and look at the simple cubic, the body-centered cubic, and the face-centered cubic cells. This link here will take you to a good summary of these with some really nice illustrations. What I really want you to focus on is this right here. You do not need to worry about the edge length in terms of R. That is not really important. And you don't need to memorize the exact numbers here, but what I do want you to know is that the simple cubic cell, it has the least packing efficiency, and the face-centered cubic cell has the greatest packing efficiency. So here is our simple cubic cell. So you can see in this illustration that we have a coordination number of 6. So this atom is in direct contact with 6 other atoms. So you can draw a straight line to the other atoms, and that's direct contact. So each of these is in direct, except for the ones on the very ends, are in coordination with 6. So that is our coordination number. And then to get the atoms, you do what we talked about on the previous slide. You look at, there's 1/8 of an atom in each corner, and there's 8 corners. So 1/8 times 8 gives you 1 atom per unit cell. Now, if we look at the body-centered cubic unit cell, we see that our coordination number is 8. Now, I want to point something out. These are not different particles. These are the same. So this isn't like sodium chloride that we're looking at. The reason they are different colors is so that you can see the difference, but these are the same. So this purple guy right here, we can draw a straight line to 8 unique other particles. And so our coordination number is 8. And then our atoms per unit cell, we still have the 1/8 at every corner, but then we have 1 big atom in the center. So you can see that this is much more closely packed than the basic unit cell that we looked at. The face-centered cubic unit cell has a coordination number of 12, and it has 8 times 1/8 and half times 6 atoms, so that's 4, because you get a half atom at every face on the cube, and there are 6 faces in a cube. And then you have 1/8 of an atom at every corner, and there's 8 corners. So you add all of this up together and you get 4. Notice how this is the closest packed structure. It has the least amount of empty space. It's about 74% occupied, I think, is what I remember from the previous slide. So this face-centered cubic cell, much more closely packed. And again, these are the same compound. These are the same particle. And there are different sizes slightly here to show you depth. But if you look here, they're the same size. And the only reason they're different colors is so that you can differentiate from the ones that are like inside the cube versus on the corners of the cube. But this would be representing the same particles. So in ionic compounds, the smaller ion will fill holes. So here is an example of 2 ions. So again, remember how I said in that last one that wasn't sodium chloride? So imagine that these here are all chloride atoms, and then the sodium atoms are going to fit in the spaces. That's kind of what we're talking about. So ionic compounds, the smaller ion fills the hole, and so there's these voids. So here you have your simple cubic structure, and there's a nice void in the middle, and so another ion can sit in that void, inside that hole right there. And so again, this is our simple cubic structure, and the ion sits there in that little void. The triangular arrangements of these three atoms are shown, and we're going to demonstrate what a tetrahedral hole looks like and what an octahedral hole looks like. So we need to draw the location of the additional atoms that would be needed. So this is the bottom of our structure. So try and visualize where, and maybe sketch in your notes like dotted lines showing where you think they would line up. Are they going to line up exactly on top? Will they be offset? Will they be switched? Try and create a tetrahedral hole and an octahedral hole. Now, if you cannot do this, that is okay. It's good if you can. It's a good exercise. However, I personally struggle very, very much with these types of exercises because I'm not very good at taking a 2D object and rendering it in 3D in my brain. So I really struggle with this kind of stuff, and I have to go very slowly and look at a lot of models. And so if you have to do that too, that is totally okay. I'm not going to make you sketch a whole bunch of these on an exam or a quiz. So here we are going to look at how we would get our tetrahedral hole and our octahedral hole. So here's our triangular arrangement of our three atoms, and the black are on the bottom, just like they were on the previous slide. And our additional atoms, so they're shown in dotted in blue on top. So our tetrahedral hole would be created by one atom sitting on top of the three right in the center. So we'd have a tetrahedron in the middle, and a little atom or ion could fit in the middle. If we want to have an octahedral hole, what we'll see is that we have them just flipped, right? So we have one, it looks like one triangle here and then one triangle here. So there's an octahedron in between the two, and you can kind of see the sides, like there's the blue, black, blue, black, blue, black. So you get this octahedron. So this will become important later when we look at our, when we try to look at how different solids fit together. So for example, the size of the hole will tell you what other particles can fit in between. So like when we looked at our ionic compound here, these holes or these voids are where the other ion will fit. And so the shape of that hole will dictate what ions can fit in there. And we will discuss this in our next lecture.